Why don't we get started? So it is a pleasure for me to introduce Andy Camilli. So actually this Monday, Andy is going to be celebrated for his 25th year at Tufts. So that dates back to 1996, I think, something like that.
It started in 95, but
95, yeah. And then, but I met Andy, and Andy's heard this story before. I think in 1990, when I visited University of Pennsylvania and was with Dan Portnoy, and Dan showed me his lab, and in his lab was this kid who looked like he was about 14, and Dan said, "This guy is really good. You should watch. You should keep your eye out for him because he's really good." And then he, after doing a fantastic thesis project with Dan, went on to a great postdoc with John Mechalanos at Harvard, and working on cholera, which he's going to talk about today. But when he, to my mind, an important event was when he decided to work on strep. And the reason is that I immediately, when I started working on strep, I sent my student to his lab to learn how to handle strep. And actually Tim Venopinen, who's now at BC, tutored my first student in strep about how to do stuff. And I was so enamored with that work that I actually moved my lab from UMass to be next door to Andy. And so now Andy's right next door to my lab, and I'm his chair, so I can demand of him any help I need on my strep project. So at the, I actually did my graduate work in phage, but I have to say I've been, for years and years, I was a very big skeptic of phage therapy, and Claudette can attest to that. But I think things have really turned around, and Andy's got some really interesting stories that's on Phage Prophylaxis, and his company, which is doing very well. And so it is actually really very fun for me to hear the story and sort of see the evolution of not only the story, but of course, it's always great to see Andy and his work.
thank you, John. I take it people can see the slides.
Yes, yes, we can.
Okay, so welcome everyone, and I'm happy to give this talk and chat about phage therapy. I actually have put in, I don't have that many slides, so there's plenty of time to ask questions and talk about things. So I'm going to start off with just a little review of phage therapy in the U.S. and Europe as it stands at the present, and then I'm going to get into my. It's not phage therapy; it's phage prophylaxis, and give you kind of some of the basics of what one does to set up a phage cocktail for, in this case, preventing an infection. So first I have to start with this. As John mentioned, I founded a company called PhagePro, and luckily we got incubator space right next door, so it's quite convenient. And they're basically working on product development, how to get phages into tablet form and that type of thing. So just a brief introduction. So phages, of course, are viruses, but they're specific for bacteria. And as far as people know, they have no ability to infect eukaryotic cells. They've co-evolved with bacteria for a long time, and that's what they infect. So this is the typical life cycle of a phage. And I should mention there's two types of phage. One kind will integrate its genome into the bacterial genome and go quiescent. And I'm not going to talk much about those phages today. I'm going to talk about the other type of phage, the virulent phages, which purely have this lytic life cycle. So they'll bind some receptor, inject their DNA or RNA, if it's an RNA phage, take over the cell, make more phages and lyse. So that's, and you can see this as a plaque in a lawn of bacteria. And wherever there are bacteria, it's phages. Typically, the phages outnumber the bacteria, several-fold. Like any good virus, the phage needs to have some receptor on the surface of its host cell that it's going to use to bind. And that's the first and most important step in the kind of determining what is the host range of a phage. And I should mention that almost all phages are very specific for a particular species and even particular strains within a species. So for example, T4 will use a particular type of LPS as its receptor on E. coli. Some phages will use surface proteins. In this case, I have a gram-negative shown here. So this would be a porin. Some phages will bind to pili or fimbriae sticking out of the cell. And then some phages will use the flagellum. Pretty much any type of structure on the surface can be used as a phage receptor. And then the phages undergo, other events to inject their DNA across the cell envelope into the cytoplasm. So, bacteria and phages have been co-evolving for a billion years, more probably. And of course, the bacteria can evolve to be resistant to a phage. And we call those escape mutants. And the easiest way to mutate to become an escape mutant is to turn off the receptor or modify it in some way that the phage can't bind. And this will come up throughout my talk. There are other ways bacteria can become resistant to a phage, but mutating or turning off the receptor is a common way. So before I get into my stuff, I just wanted to, because there's some clinicians on the line, I thought I'd talk about what's happening in the US and Europe right now. And really the model is this personalized medicine, compassionate use model. And there's only about a few dozen clinical reports, and I'll mention a few examples in a few moments. So this is specific to patients, usually to patients that are chronically infected with the multi-drug resistant bacterium. And so the patient, the physician, the family, they're desperate for something that can treat the patient. And typically these patients are going downhill at the time compassionate use of phages is attempted. So the basic protocol for these cases is you isolate the pathogen from the patient. You screen for a couple of phages. I've seen cases where they used one phage and it worked. They cured the patient. Others typically go with a cocktail. And so they'll screen for phages from collections that can kill the strain in vitro. And I've listed a few examples of where collections can come from. They can come from individual academic labs that study pathogens. A couple really big banks of phages exist now. So the Naval Medical Research Center has a huge bank of phages, and they've been involved in several compassionate use cases. There's some companies and examples, Adaptive Phage Therapeutics, they have a very large bank of phages. And I'm going to talk about one case report where the phages came from what's called the C-Phages program, which is basically hundreds of high school students around the country have been isolating phages from the soil, wherever they live, on this Mycobacterium species, Smegmatis. So anyway, you screen for phages that kill the strain from the patient. You purify the phages, and then you get an emergency use authorization to treat the patient. You treat the patient, and you see what happens. I should say that often the treatment is done in conjunction with antibiotics, even though the antibiotics have not been successful. Often they keep the antibiotic pressure on the pathogen when they administer the phages. And if the patient gets better but then relapses, they will isolate, they'll repeat this, isolate the bacterium, and typically an escape mutant has arisen in the patient. So now they need new phages or phage mutants that can now kill this escape mutant, and they repeat this. So there have been about 40 clinical reports since 2008. These are in English, in English journals. There have been a lot more in other languages. And overall, I've heard this said, I haven't actually tallied this up myself, but I've heard it said that there's about a 70% success rate amongst these reports. Now, and here's some examples before I talk about the grain of salt. So here's a few, a really famous example was this case in 2000, I think it was published in 2017, was an Acinetobacter baumannii infection of a patient. And that was done by, it was a collaboration between Schooley and the hospital and some other labs to treat a patient. And I believe they treated that patient intravenously with phage. Another recent success case was this disseminated Mycobacterium abscessus. And that's where the C phages came in. That's where so this was Graham Hatfull in collaboration with, again, the clinicians. I think there was a company involved and a bunch of laboratories. And they screened a subset of phages from the C phages program. I'd mentioned that these phages were isolated on this non-pathogen, Mycobacterium smegmatis, but they found a few that could kill this different species, abscessus, and treated the patient successfully. Some other examples are a Klebsiella pneumoniae biofilm-associated infection. This used a cocktail of phages. And then here's a few more examples that I gleaned from the literature. Now, I have here this salt shaker because these are basically a collection of N's of 1, if you think about it. There's been very few clinical trials, and I think it's hard to design a clinical trial when the patients are, when it's, compassionate use type of situations. There are some clinical trials ongoing right now, and there have been a few clinical trials that were completed over the last couple decades, and a couple really big ones have failed. So it's not quite clear what the future of phage therapy holds. But from my perspective, these N's of 1 are often pretty convincing. For example, this first case, the patient had been treated for many weeks and was not responding to treatment. And as soon as they gave the phage therapy, the load of the bacteria dropped, the patient got better. And then a few days later, he relapsed, and they isolated the bacterium and it had become resistant to their phage. So then they treated the patient with different phages that could now kill this escape mutant, and then they clear the infection. So when you see cases like that, it's pretty convincing that the phages actually can be attributed to the success of the treatment. Okay, so that's phage therapy. Now I'm going to switch to what my lab's working on and my biotech company, and that is phage prophylaxis for cholera. And it's really, we've done the basic research. We haven't done any clinical trial yet. I'll talk about that at the end. So just a brief introduction to cholera. It's this highly transmissible disease. As you know, patients get rice water stools and vomiting, and people will die by dehydration, and rapid rehydration therapy is critical. And there's millions of cases, et cetera, et cetera. So this is caused by this gram-negative bacterium, Vibrio cholera. This is an aquatic organism. You find it in temperate ocean and estuaries all over the world. Only some of the strains have cholera toxin and are pathogenic. And wherever you find this bacteria, you find lots of phages that specifically can kill it. Over the last decade, Vibrio's become really drug resistant, MDR, even XDR in Africa and in India and in Bangladesh. So it's really becoming a problem. So briefly, let me mention that there are ways to protect from cholera, and that is vaccines. So it's been known for a long time that patients that survive cholera have long-lasting, high-level immunity. The dominant protective antigen is the O1 antigen of the LPS. So the O1 serogroup is the predominant cause of cholera, and it's specific sugar in this O antigen, and that is protective. So there's a couple whole cell killed oral vaccines that can give partial short-lived immunity, and these are in use. Typically, they're used retroactively or in clinical trials. They're not really being used in a widespread manner proactively. A lot of that has to do with limits on how much vaccine's around, cost, etc. There is recently a live attenuated vaccine called Vaxchora that was approved. It's in the US, it's approved as a traveler's vaccine. And in the clinical trial, it was shown it gives high level immunity, but they only tested it out for 90 days. So it's of unknown duration. But this is looking like a really good vaccine, this live attenuated vaccine. Now, one of the problems with any vaccine is it takes a while to take effect. So the oral vaccines take about 14 days to take effect. You need to have a boost, ideally 2 boosts. And I'm going to talk about why that's a problem in a second. So this is the traditional life cycle of cholera. There's contaminated water. The susceptible person ingests the water. They get cholera. Out comes the bacteria and the profuse secretory diarrhea, further contaminating the water supply from which other people get sick. However, it's become more and more appreciated the last couple decades that actually most cases that occur during an outbreak, at least in crowded areas like Dhaka or refugee camps, whatever, et cetera, is not this mode of transmission, but rapid transmission within households. So a couple studies have shown that the rate of infection during an outbreak goes up about 100-fold if you reside within a household where somebody comes down with cholera. And the peak incidence of these secondary cases is about 2 to 3 days after the index case presents. So what's happening is the cholera is spreading in the household. It's not really known how. Is it fomites? Is it direct contact? Is it contamination of the water in the household? But however it's happening, this is a huge problem. So it's been estimated that during an outbreak or epidemic, about 80% of cases are due to household transmission. Now, the problem with this rapid transmission is you can't intervene with the vaccine. The peak incidence is 2 to 3 days after the index case. You could give prophylactic antibiotic therapy, and that has worked in the past, but is discouraged now by the WHO and others because of the effects on the host microbiota and also the emergence of multi-drug resistance. So this is a gap that needs to be addressed, and that's where we're thinking of applying phage therapy. Could we give the people in a household where there's been a case of cholera a cocktail of phages and protect them? So where can one get phages? Well, you can get them from the environment, but what we've been doing is just isolating them from cholera patient stool samples. So this rice water stool that cholera symptomatic patients have is loaded with Vibrio cholera. There's about 10 to the 8th CFU per mil, and there's aggregates in this mucus. And a typical adult will end up shedding about 10 to the 13th Vibrios back into the environment. Now, some rice water stools contain phages. We can just isolate them from this liquid and see them as plaques in a lawn. And this is not a new discovery. The co-discoverer of phages over 100 years ago, Felix Durel, was a physician from Canada doing work in Europe, and he discovered phages in cholera patient stool samples. So we kind of picked up and started applying, modern molecular techniques and more, reproducible controlled experiments. So kind of the first experiment we did with one of these phages isolated from a patient was done by Eric Nelson, who was at the time an MD PhD student in my lab. He's now has his own lab at University of Florida. And what he did was he just asked if we add phage to the inoculum just right before we inoculate mice, can we reduce the load of Vibrio cholera in the small intestine? So this is the infant mouse model. You slip a little tube in there and put the bacteria into their stomach. It goes into the small intestine and the bacteria will colonize and multiply. Now, if you don't give any phage, all of the mice are infected and the load is pretty high, about 10 to the 7th CFU per small intestine. But as Eric added increasing amounts of phage to the inoculum, these are different groups of mice, he could knock down the load of the bacteria after 24 hours in the small intestine. So this is the multiplicity of infection. So here is 1 phage per Vibrio added to the inoculum. So this isn't really phage therapy. Well, it's kind of a borderline between phage therapy and prophylaxis because we mix the phage right before we instill the bacteria into the mice. So with this success story, we kind of went on and thought more about how can we do this. And so we came up with a couple of questions that I think are general for thinking about phage therapy and phage prophylaxis. So in our case, are there Vibrio-specific phages that are active in the context of infection? Now, if you show a phage can kill a bacteria in the laboratory, it doesn't necessarily mean it's going to work in whatever organ or tissue is being infected in a human. If there are such phages, what's their biology? What's the host range? What receptor are they using, et cetera? What are the mechanisms of escape from the phage? And this, I'm talking about mutational escape. And any bacteria is going to become resistant to one of its phages. It's inevitable. So if that happens, what's the frequency? And importantly, for what we're studying is, do escape mutants remain infectious? And I'll talk a lot about that today. And then finally, if you get through all these three, can you put together a cocktail of phages, in our case, that could prevent cholera in animal models? And if so, will the phages disrupt the gut microbiota? So I'll address all these things. So in order to do all this work, we've collaborated with people in Bangladesh. So this is if you've ever been there, it's a beautiful country, incredibly nice people, but it's situated in the Bay of Bengal, and it has these huge rivers flowing down from the Himalayas, and it has these tremendous areas of estuaries where, twice a day with the tide, the salt water rushes in, mixes with the fresh water, rushes back out, and there's a huge population of people living down here. Now, this is kind of the home of Vibrio cholera. It likes estuaries, and the toxigenic strains that have cholera toxin like people. And so there's a lot of cholera here. There's epidemics twice a year that they begin in these estuaries and move inland, and typically Dhaka has these big outbreaks. So I've benefited for 20 years now from collaborating with really talented researchers at the ICDDRB in Dhaka. For many years, Firdousi Qadri and I collaborated, and we published some nice papers together. And in the last few years, I've been collaborating with Munir Alam. And again, they're both in Dhaka. So we started by first trying to collect some phages. And this was work done with Firdousi Qadri and her lab and my postdoc, Kimberly Seed. And basically, we just asked the question of, are there phages coming out in patient stools? If so, what are these phages? So we took advantage of a collection of glycerol down rice water stool samples that Dr. Qadri had in her freezer. And Kim took 3 different stools per year, thawed them, and screened for phages. We looked for plaques on a lawn of Vibrio cholera. You can see we have the sporadic plaques coming from these different patient samples, and we could isolate the phages, and we sequenced their genomes and found that there are 3 new phages that have never been reported before. We call them ICP1, 2, and 3. And we've done a lot of work on these. I won't bore you with the details, but we know a lot about their genomes. We've determined their receptors. So ICP1 and 3 use the O1 antigen as a receptor. Interestingly, the 2 phages are using it in different ways because we can get escape mutants that have mutations in O1 synthetic genes that are still susceptible to ICP3 and vice versa. So we don't know at a molecular level what's happening there. And then we have this other phage, ICP2, that uses an outer membrane protein, a porin opiate that I'll talk about. So these are the three phages we found, and we haven't found other phages, even to this day. Now, once we had these three phages, Kim designed PCR primers to go back and screen these stool samples. And what she found was pretty interesting. ICP2 and 3 were still kind of sporadic. ICP1 we found in every stool sample. So it's very frequently found in stool samples in Bangladesh. Now, we don't find these three phages everywhere in the world. For example, ICP1 and 3 we did not find in Haiti, but we did find ICP2 there quite frequently coming out in cholera patient stool samples. ICP1 we also see in Africa and in India, but not ICP2 and 3. So that's kind of interesting. Now, you might be thinking, why do we have PCR positive stool samples, but we don't see plaques? Well, the reason is because sometimes the patient is shedding escape mutants. Even though the phage is present in that stool sample, there's been a selection in the patient intestinal tract for Vibrio mutants that can escape the phage that was just killing it. And I'll talk about this in a moment. Now, the first thing we did is to try and find the receptor. Is you just grow the bacteria up in the lab, add a phage, add one of these phages, and ask, do you get escape mutants? You can then sequence their genome and ask, where's the mutation lie? And that hopefully will tell you what the receptor is. So for ICP1 and 3, we very quickly get escape mutants that are resistant to these phages. In whole genome sequencing, again, this is work Kimberly Seed did, revealed mutations in 2 genes that are part of the O1 antigen biosynthesis loci, so WBEL and MAN-A. And what she found is these were frame shift mutations that knock the gene out of frame. And the frame shift mutations were happening in these poly-A tracks. You can see this gene has 1 poly-A track, MAN-A has 2 poly-A tracks. Now, what happens is during replication, at a pretty high frequency, the bacteria will make a mistake and either add or subtract an A. This is called slip strand mispairing during a DNA replication. And this puts the gene out of frame, and thus they're not making the O1 antigen, they become resistant to the phage. This is a type of phase variation that bacteria often use, and it's a reversible process. So this mutant can give off a daughter cell later on that adds back the A and puts the gene back in frame. So this kind of rapid on-off switching allows the bacteria to become resistant to the phage. Now, we don't see, I mentioned we got these phase-off mutants in vitro. We don't see these phase-off mutants coming out of humans. Why is that? Well, it's because the O1 antigen is a critical virulence factor. Matt Walder showed that many years back. And you can test this in our infant mouse model. So here we took some of these frame shift mutants, the WBEL and the MAN-A, and we did competition assays in the infant mouse model. So you just mix it 1-to-1 with a wild type strain, infect the animals, 24 hours later, you ask what's the ratio of the 2 strains in the small intestine. So this O antigen off mutant is outcompeted by the wild type over 1,000-fold. And the MAN-A is also attenuated. We can revert these mutants back and they regain virulence. So this is why we're not seeing these phase off mutants coming out of humans, is because they're attenuated. But I just told you that often we see Vibrio coming out of patients that are escape mutants of some sort. So what are they? Well, what Kim found is that there's this mobile island called a plea that can give resistance to ICP1, and it's very specific to ICP1. And roughly 15% of strains in circulation have it, and it can move around by horizontal gene transfer. This 18 kb island has a bunch of genes, And I won't go into the details, but basically upon ICP1 infection, this locus will excise from the genome and replicate as a plasmid. And so Kim tested this, she showed this by designing these outward facing primers. These will not yield a PCR product on the genome, but if this element pops out of the genome as a circle, these will now give a PCR product. And what she showed is that within 5 minutes of infecting the bacteria with ICP1, you see this excised circle, and it replicates to high copy number and gives resistance to ICP1. It's not induced by other phages, so it gives immunity specific to ICP1. And we found 5 different versions of this plea in 4 in currently circulating strains, and 1 we found in a strain from the 1930s. So this is a way of having resistance to the bacteria. It has nothing to do with mutating the receptor. And Kim's lab has been, they've published a few papers over the last few years on how this element works, and it's pretty interesting stuff. I'm not going to go into that, but basically if a Vibrio has the plea, and you look at plaque-forming units or replication of ICP1, it's flat. The phage is not replicating on this plea1 host. If you have an isogenic strain that's missing the plea, the phage infects, replicates till it's basically killed all the bacteria in the culture. And you can also look at the average phage burst size on a plea minus host, and basically each cell will liberate about 90 phages when it lyses, but strains that have the plea are protected. So this is a paper out of Kim's lab, not my lab. So this is a little taste of the arms race that can happen between phage and Vibrio, and it's something we have to be aware of if we're going to go in and try and use phage prophylaxis or phage therapy. Now, what was interesting is we thought we had things figured out at this time for ICP1 and 3, but another discovery happened, and that is that Kim occasionally could find an ICP1 phage that could still form plaques and kill a plea containing Vibrio. So here's an example. This Vibrio host in the surface of this agar plate has a plea, but here was an ICP1 from patient that could still form plaques on it. And we were perplexed at what was going on. And so again, whole genome sequencing came to the rescue, because when Kim sequenced these particular ICP1 phages, she discovered that they contained a CRISPR-Cas locus that was missing in other ICP1 isolates. And it turns out this CRISPR-Cas is present in about 20% of ICP1 phages that we've isolated from Bangladesh and from India. So you're probably familiar with CRISPR-Cases as a defense mechanism bacteria have to protect against phages. So here the tables have turned. This is a phage-encoded CRISPR-Cas. And as far as I'm aware, it's the only CRISPR-Cas found in a phage so far. So this is the Cas locus in the phage. It's got 2 CRISPR arrays. And upon sequencing, we immediately figured out how this thing works, because all of these spacers that will make guide RNAs in this green color here are targeting pleas, one of the pleas. And this particular phage isolate from 2011 has spacers targeting all 4 currently circulating plea elements in Vibrio cholera. So this is a good phage to have in our cocktail, because it can defeat all of the pleas currently in circulation. This CRISPR system is fully active, because if we destroy, so this 2011 isolate, spacers 8 and 9 target 2 genes in the plea1 that I'm showing here. And what Kim did is she went and destroyed these 2 spacers. So now the phage couldn't target the plea with its CRISPR, and she could isolate rare phages that had picked up new spacers, and they mapped all over this plea element. And so it really confirmed the model that the phage uses its CRISPR-Cas to attack the plea. And this is kind of a slide I put together adapting this figure from this review, where When ICP1 infects Vibrio cholera, it injects its DNA genome, and the plea is very quickly activated, as I showed you, within 5 minutes to excise, replicate, express its genes, and it blocks ICP1. But if ICP1 has a CRISPR-Cas system, it's a race between the two because it can make the guide RNAs and the Cas proteins and degrade the plea. And so if the phage has the CRISPR-Cas, it defeats the plea and can lyse the cell. So this is another arms race that we're glad we figured out because it helps us think about the phage therapy in a more productive way. So, so I'll just talk briefly about one other phage, ICP2. This one's sporadically present. It uses a protein as its receptor. What is that protein? It's called OMPU. It's the major outer membrane porin that's made during infection. This is interesting. During growth in the laboratory, Vibrio only makes a little bit of it. It uses another porin during growth in the lab. But during infection, expression of OMPU is upregulated highly, and it's because this is a critical virulence factor. It's co-regulated with cholera toxin and the pili that are needed for colonization. And the master regulator of all these genes is toxar. So we found it interesting that this phage is using a receptor that's upregulated during infection. It kind of feeds into our working hypothesis that these three phages have evolved to tag along with Vibrio cholera into cholera patients, into humans. And when Vibrio colonizes the small intestine and grows to high titers, the phage can then bloom on the bacteria. So it would make sense that the phage would evolve to use these essential virulence factors, and in particular, one that's upregulated during infection. So I mentioned this as a critical virulence factor, and yet we still see escape mutants coming out of some patients that have cholera. So I say here in this title, some cholera patients are shedding escape mutants that are resistant to ICP2 that have null mutations in toxar. I just mentioned this is the major kind of master regulator of the virulence factors. So in essence, these patients are shedding avirulent mutants. And we showed that. We took some of these toxar null mutants isolated from cholera patient stool samples, did the competition assay in infant mice, and they're highly attenuated. So again, these patients are shedding, the phage pressure on the bacteria is so high in the intestinal tract of these patients that they're shedding these avirulent mutants. Kim Seed and I showed this in our eLife paper a few years ago. Now, if you think about phage therapy and phage prophylaxis, what I'm showing you kind of bodes well for doing phage therapy or prophylaxis in that these escape mutants are attenuated. But we sometimes see escape mutants that are not attenuated, and that is going to be a problem for phage therapy. So we found that some patients, just a few, but some, were shedding escape mutants where there were these single amino acid changes in OMPU, specifically in these two outer loops. Turns out the recent crystal structure showed that loop 4 and 8 are actually together in this mature trimer porin structure. And so what we think is the phage tail fibers are probably binding to this region, and that these mutations are blocking the phage's tail fiber from binding. So we recently confirmed that by work from a graduate student, Andrea Lim, who just left for a postdoc. Basically what she showed was she selected for phages that would regain the ability to plaque on these escape mutants. And when she isolated lots of ICP2 phages that could now plaque on these OMPU mutants. And what she found is that she was getting mutations in these two phage tail fiber genes. I don't have time to go into it, but there was kind of an order to which they arose. But the main point here is the phage can evolve back and regain the ability to plaque on these mutants. So now back to the phage therapy, phage prophylaxis idea here. So I mentioned earlier, there's this problem of household transmission. So can we come up with a product that might be able to protect the household contacts or anyone at high risk? It could even be medical workers during an epidemic. So we started testing a phage cocktail made up of these three phages, ICP1, 2, and 3, in our two animal models. I mentioned one is this infant mouse model of colonization. We also have an infant rabbit model that they become symptomatic. So this was work spearheaded by two former trainees in my lab, Mimi Yen, who now is running PhagePro, this biotech company that we founded, and a former postdoc, Lynn Cairns, who's now a medical writer in England. And what we did was we tested out the cocktail in first the infant mouse model. So this is the experiment. We gave 10 to the 7th phages, 1-to-1-to-1 mix of the three phages to the infant mice. We waited 3 hours, so this is given at T-3 hours. We then challenged the mice with, this is roughly about 10, anywhere between 10 to 100 times in the ID50 of Vibrio cholera. We then measured the load of Vibrio cholera in the small intestine at 24 hours. So again, if you don't give phage, all the mice are infected and there's a high load in their small intestine. And what we found is individual phage could give varying levels of protection, but the cocktail did the best. ICP3 alone and the cocktail protected most of the mice. So this was a good result. It told us, yeah, we can at least 3 hours prior to a challenge, we can protect the mice. So this encouraged us to ask, well, how long did the phage hang around in the small intestine? So here we gave the phages individually to mice, and we did not challenge with Vibrio cholera. This is just asking how long do the phages stick around in the small intestine. And basically what we found is that 2 of the phages are retained up to 24 hours. You know, it's going down a couple of orders of magnitude, but they're still there. But really, ICP3 is really starting to drop out by 24 hours. This was the maximum length of time our ICOOK protocol allows us to do the experiment. But we liked it anyway because we thought, okay, well, if there's a similar retention in humans, we can give the phage cocktail once a day. So this spurred us on to test for phage prophylaxis at longer time periods between giving the phage and challenging. So in this experiment, we tested 6, 12, and 24 hours of prophylaxis. And again, what you can see is high load without phage, and we start to see increasing colonization as we wait longer and longer, and there's this kind of bimodal protection at 6 and 12 hours. At 24 hours, all of the mice are infected. But I point out that this is over an order of magnitude lower than the non-protected mice. And what's interesting is in these mice that have some kind of a smoldering infection going on in them, we isolate lots of these Vibrio cholera in their intestine are escape mutants. And when we sequence them, we see that they're typically O antigen off mutants that are attenuated. So now the problem here is we can't, so the prediction here is these mice have this smoldering infection of attenuated Vibrio cholera, but the infant mice don't really have symptoms of cholera, so we can't really look at pathogenicity. So we switched then to the infant rabbit model. This was a model kind of perfected by Matt Waldor's lab a few years back. And so it's a bigger animal model, so we give more phage. We waited 3 or 24 hours, and then we challenged the rabbits with a high dose of Vibrio cholera. This again is about 100 times the ID50. And then we measured the load of Vibrio cholera in the intestine. So what you can see is without any phage prophylaxis, the rabbits have a high load in the small intestine. And these rabbits are very sick. They have profuse secretory diarrhea, and they rapidly lose body weight. And we euthanize them once they've lost 10% of their body weight. And in fact, all these animals had to be euthanized quite early in the experiment. Now, the prophylaxis groups, the 3 and the 24 hour, we saw this bimodal protection at 3 hours. And similar to the mouse model, at 24 hours, all the rabbits were infected. But here, the load is about 2 orders of magnitude lower than these mice, or sorry, these rabbits. And again, in these rabbits, we isolate most of the bacteria are escape mutants that are avirulent. They've turned off O antigen expression. So again, what we're seeing is this kind of smoldering infection where most of the bacteria are avirulent escape mutants. I'm not showing the data here, but the titer of the phage is very high in these rabbits because as the bacteria replicate, the phage can replicate. The important question here is, well, are these rabbits symptomatic? And the answer is no. None of these animals had profuse diarrhea. They didn't lose body weight. And I'm showing that here in this body weight at end of the experiment. So if you have the mock group, no phage, no challenge, here's their body weight. Now you'll notice they're all losing a little bit of body weight because they're not, the pups are not with the dam throughout this experiment. And we have to do that because the doe will kill the pups, the infected pups. The no phage prophylaxis but challenge group all lost a significant body weight, and these were all euthanized early in the experiment. These all went the full 24 hours after challenge. None of them had symptoms, and there was no significant body weight loss compared to the control group. So it appears the phage cocktail is working in these two animal models, and we're hopeful down the road, if we can test this out in a clinical trial in humans, that we'll see a similar protection. But the last bit of data I want to show you is the question about, will these phages affect the gut microbiota? I mentioned phages are usually very specific for their host bacterium, but we wanted to test it. So we use an adult mouse model here where we gave the cocktail to mice. We had a heat-killed phage cocktail control group, so the phages are killed and shouldn't infect. And then we had our positive control, vancomycin. And we basically just looked at changes in the gut microbiota over time. What I'm showing here is a rapid change in the microbiota in the antibiotic-treated group. So before antibiotics, the microbiota was over here, and it just rapidly goes over here. But our heat-killed control and our live phage group, there was no change. All of these microbiotas are all grouped here in this cluster here. So as we had predicted, our phages, are not killing gut microbiota species. So let me just kind of sum up and do some acknowledgements, and hopefully you'll have some questions for me. So I showed you that there's 3 phages that dominate as predators of Vibrio cholera within cholera patients, and we've been doing basic research on these phages and found they all use receptors that are essential virulence factors. And this is good for phage therapy and prophylaxis because it's really going to limit the ability of the bacteria to escape the phages by just mutating the receptor. ICP1 has another way to, Vibrio cholera has a plea defense system against ICP1, but in a really cool example of an arms race, the phage has a CRISPR-Cas that can overcome this plea defense system. So our phage cocktail contains a phage that can rapidly change. It can acquire new spacers and keep up with the bacterium. And this is something we're excited about. We're not sure what the FDA is going to think about a rapidly changing phage, but we'll see in the next year or two. And then I showed you that a cocktail of these 3 phages can work prophylactically and prevent cholera symptoms in animal models, and that the phages are specific for Vibrio and don't affect the microbiota, at least not of mice. So I mentioned my collaborators in Bangladesh who've been critical to this, isolating these phages and doing a lot of these experiments. The work in Bangladesh has been done for many years in collaboration with Stephen Calderwood and Jason Harris, who are here in Boston. We did some work in Haiti that I didn't show you. I showed you a little bit of work from it, and that was done in collaboration with Jacques Bouncy and Luis Ivers. And then importantly, the people in my laboratory that did a lot of the work I mentioned all these people, current people in the lab that are studying phages and Vibrio, and some other organisms are listed here. Liz Tan, Isaac is he's a new grad student. Kyle's a new postdoc, and Dave Lazinski's had a hand in pretty much all the work that I showed you today. So thanks for your attention, and I'd be happy to answer any questions.
Great. So thank you. That was really interesting. So can I start off with the first question?
Please.
So you found that at least one of the phages seems to hang around in the mouse gut longer than others. And do you know why? And might there be a way of selecting for phage, which are particularly hardy in that way? And I don't know if it's acid resistance or if they attach to some host epithelial something. And might that increase the effectiveness of the cocktail?
Yeah, that's a good question. We don't know why the phages are retained so well and why they work in these animals. I should mention that we've tested another phage that doesn't work in these animal models. So not every Vibrio phage is going to work. We don't know why they're retained and why one of the phages isn't retained so well. Some phages that are found in the human gut have, for example, lectins on their surface that let them stick around on carbohydrates on intestinal epithelial cells, mucus. We don't find, at least by annotation, we can't find lectins on our phages. It could be other properties of the phages that allow them to stick around. Since we find these phages very frequently in humans, we're hoping that they have a long retention time in humans as well. We're avoiding isolating mutants or engineering the phages in any way because we want to be able to go to the FDA and authorities in Bangladesh and other places and say these are natural phages. We haven't altered them, so we haven't done such a selection, but we could if needed down the road.
So I should.
Yep.
Go ahead.
No, I was going to move to James, but let's finish, Sean. Go ahead.
Yeah, no, yeah. So I was just going to say, please put your hand up in the chat or whatever to be able to ask a question. Please, James.
So that was super fascinating. And so as an engineer, one of the first things that came to mind for me was, okay, so like these are amazing natural starting points that you have here, but what would be the prospects for engineering these phage? And it sounds like there are regulatory hurdles that might be issues to consider in the future. But I guess, if we're sort of discounting those, like how easy or difficult is it to think about trying to improve the properties of these phage as therapeutics? Like, is that a totally crazy idea? Like, is it moderately crazy? Like, what can you say about the prospects for engineering phage for therapeutics in the future?
Yeah, I mean, it's for many phages, it's very easy to manipulate them genetically. So Kimberly Seed set up a way to edit the phage genomes very efficiently. And so we can pretty much do whatever we want to these phages. And so that, and also people are now doing synthetic phage engineering where you can basically, design a phage in the lab. I think we benefit from having these phages that naturally have evolved to prey on Vibrio in the human intestine. So nature's done the job for us. I think our, in terms of engineering would be things like what John just mentioned. Can we get them to stick around longer in the intestine? Might we find mutants that are more resistant to passage through the stomach? Can we enhance the repertoire of phages to include, for example, ICP2 phages that can already prey on some OMP-U mutants that commonly occur in patients? That is also going to be a problem with the FDA because it's already a 3-component product. And if we start adding in more and more phages, it becomes more and more difficult to manufacture. But I think there's a world of opportunity for engineering phages to target particular receptors. I know there's some companies out there in academic labs engineering in CRISPRs into phages so that they can target particular genes. You know, phages naturally are going to kill the host cell anyway. So, but I think there is opportunity to enhance them. I hope that answered your question, James. I think there's lots of opportunity to engineer them is my answer.
Awesome. Thank you.
Mandy.
That was a great talk. Thanks. So obviously I'm interested in all the mycobacteriophages, and I was just wondering if you could speculate on how possibly that would be administered in the context of trying to treat like multi-drug resistant tuberculosis. Would it have to be some sort of nasal, the administered phage?
Yep. Yeah, great question. So yeah, I didn't mention that, but cholera is kind of a low-hanging fruit. It's extracellular in the lumen of the intestine, so we can just give the phages orally. But that's clearly not the case for those other pathogens, many of them that I mentioned at the beginning of the talk as examples. If you look at all the case reports where phage therapy has been used and published, about a third of them were intravenous, about a third were topical. I think the topical ones are really going to be hard to interpret, you know, what's happening there. But roughly 10–15% were given as aerosol, or even dry down phages given intra-lung in a kind of a spray. And there were a few other routes of administering the phages. So there have been lots of different routes of administration. I think you can get the phages to different organs and tissues. I think for intracellular pathogens, it's a different ballgame. So for example, there are extracellular Mycobacterium tuberculosis cells, but they're also intracellular. And I doubt you can get phages inside a macrophage, for example, and kill the organism inside the bacteria. But maybe just having pressure on the extracellular bacilli would have an impact. So phages have been used against Pseudomonas aeruginosa in CF patients. So I think there's a lot of opportunity to administer the phage to wherever the pathogens are and give repeated doses. But again, I think the intracellular pathogens, it's another ballgame altogether.
I'm going to ask a quick follow-up question for engineering question. So to that point, do you think they could be encapsulated in some way, that in some sort of formulation that would promote the macrophages to ingest them?
Yeah, that's a great idea. You know, it's the COVID RNA vaccine, but with phages inside. Yeah, I could foresee that. There would be ways to do that. Yeah, one can make, you know, vesicles that can fuse with cells, and maybe you could even target it that way. So, but if you did that, or maybe you could target, you know, the endocytic pathway, but still, would a phage be able to work inside of a human cell? I don't know. There's certainly not in an endosome, but maybe, you know, you could deliver them to the cytoplasm, or a endosome that hasn't fused with lysosomes. Yeah, I think our imagination is there's no limit to what one could engineer.
Amy.
Hi, Andy. Thanks. That was a really great talk. Can you guys hear me okay?
Yeah.
Okay. I had a couple of questions. When the phage develop this resistance, or and can propagate, but then they lose virulence, do you think those phage now have, like, are they able to propagate in the environment? Like, I'm just sort of wondering about the ecology or the ecological dynamics of, you have like people that are infected, and then, you know, like producing phage that are no longer really good at infecting humans, but now able, you know, like, I don't know what happens to those. Are they complete dead ends, or do they have like a second life? Like,
Yeah, that's a great question. So we looked at that a bit. So the OMPU point mutants that escape ICP2, we don't see them. If you look at, the hundreds of clinical isolates that have been sequenced, you don't see them. We see them because we sequence dozens of single colony isolates from patients. So if you look hard, you can find them, but you don't see them. And what that tells me, because the isolates that have been sequenced and put in databases were all clinical isolates from various outbreaks over the years, what that tells me is that these OMPU mutants are not surviving in the environment. And we actually did an experiment. We took one of these OMPU point mutants that's still virulent, and we passaged it in the lab, and it does have a fitness cost, this OMPU mutation, just for growth. So those mutants appear to be selected against over time. The toxar null mutants, the O antigen off mutants, they're going to be highly selected against, and because they're avirulent. And I would imagine that an O antigen mutant, a rough mutant, is not going to do well in the environment either. So yeah, so I guess that's my answer, is we don't see these escape mutants amongst all the clinical isolates that have been sequenced. They seem to be transient, and only when there's a high titer of phage preying on the bacteria. In our case, it's in our animal models and in a few patients. Most patients are not shedding escape mutants. And as far as the phages, yeah, they will, these phages are naturally present in Bangladesh. And, if we give them to patients, I don't think we're disrupting the balance over in that country. The phages are there naturally anyway. I think if we start engineering the phages and they're shed, then that brings up new questions about what the phages might do in the long run.
And then I had a question about the CRISPR-Cas system, the phage one. Like, have you selected for resistance mutants in the bacteria to those?
Yeah, we don't see them yet, but the phages can make inhibitor proteins that block CRISPR systems. So Kim is actually looking at this, are there Vibrio strains that make protein inhibitors of the phage CRISPR system? And I'm not aware of whether she's found any such proteins yet. We haven't run into that yet in the few phage Vibrio strain combinations we've tested, but it's likely the bacteria have such a system. We just haven't found it yet.
Thanks a lot, Andy. It was really cool to see it all together.
Thanks.
All right, it's getting late, but I want to ask one more question, sorry. So it seemed to me that you found maybe different sets of ICP phage in Haiti and Africa. So does that mean that if you want to now do prophylaxis in Haiti, you have to develop a different cocktail?
I would, well, Haiti only had ICP2. It had another phage that we isolated and tested, and it doesn't work in mice. So it may not work in humans. So I would like to, if cholera returned to Haiti, and I had my way, I'd use the cocktail of ICP 1, 2, and 3. I can't imagine it would have some impact on causing evolution of hypervirulent strains in that nation, but it would be up to the regulatory authorities of Haiti. Would they allow us to bring in phages that are not naturally present there?
All right, so I don't see any other questions. So thanks a lot, Andy. This is great. And thanks everybody for showing up and participating.
If anybody has burning questions that they didn't get to ask, send me an email. I'm happy to chat. And thanks everyone.
All right.
Great talk, Andy.